pediatric leukemia is the most common cancer that we see in kids. There have been dramatic improvements in outcome, and you can see this incredible improvement. But there are still relapsed cases of leukemia that have not so great prognosis. And infant leukemias in particular are tumors that really we need to make more progress, and the overall survival for infant leukemias is less than 50% at this point. The work that I'm going to tell you about stemmed from a patient that I saw about 12 years ago who was a little 3-month-old baby who came into her pediatrician because she had cold symptoms. She ended up having a white blood cell count of about 450,000, which is about way higher, 50 times higher than normal. And she ended up having type acute myeloid leukemia that was very aggressive, and she died about 10 days after she was diagnosed. We were able to collect some of the cells from her leukemia, and we identified a specific abnormality in her cells that resulted in a translocation protein that fused a gene called MLL to a gene called CALM. And so on the 5' end is MLL and the 3' end is CALM. MLL stands for mixed lineage leukemia. It's a gene that's involved in many leukemias in children and adults. CALM is a gene that encodes for a protein that's involved in endocytosis, so how things get into the cell. And not that much was known about it, but CALM translocations have resulted in aggressive myeloid leukemia in infants. CALM is also fused to another gene called AF10 that's seen in both childhood and adult myeloid leukemias and about 5 to 10% of T-cell leukemias. And so in this case, CALM is on the 5' end and AF10 is on the 3' end. And again, CALM is thought to be involved in endocytosis, but the fact that it is involved in these translocations and leukemias suggests that there's something inherent about CALM that's important. Both CALM AF10 leukemias and MLL leukemias share similar gene expression profiles, and Dr. Mulligan showed you a slide similar to this that looks at the upregulation of different genes in particular leukemias. And in CALM AF10 translocations, like the MLL ones, there's high expression of genes that are called HOXA genes. And HOXA genes are very important in the pathogenesis of many different leukemias. HOXA genes are very conserved. They're essential for normal embryonic development. They're involved in the normal development of white cells. If you mess up HOXA gene expression, that can halt maturation and lead to leukemia. And what you can see here is, in the upper panel down here, normally HOXA genes decrease and cells mature. When HOXA expression is maintained, the cells undergo self-renewal and you get leukemias. So HOXA genes are associated with leukemogenesis. Now, over many years, we studied the role of CALM in endocytosis and tried to make a link between endocytosis and cancer. But a couple of years ago, we stumbled on the fact that CALM contains a nuclear export signal. So in this CALM AF10 fusion, this red bar over here is a nuclear export signal, which is a specific sequence of amino acids. In order for proteins to get into and out of the nucleus is bounded by a membrane, and there are these little purple things which are pores. And these are made up of very complex sets of proteins. There's about 30 or 40 different proteins that are involved in making a nuclear pore. And in order for a protein to get into or out of the pore, it has to actually bind to other proteins which recognize the pore and then moves it in one direction or the other. So a nuclear export signal is a short amino acid sequence, and it's about 18 amino acids. And it forms a complex with a protein that's called CRM1 or CRIM1 and another thing called DRAN-GTP. And that's shown over here. This is the cytoplasm. This is a nucleus here. Here is the receptor, which would be CRM1 in green. It binds to this cargo, and it basically gets shuttled out through this pore. Importantly, CRIM1 function is blocked by a drug that's called leptomycin B, which was identified as a product of fungi. And this sort of shows you more in detail what happens. Again, this is the cytoplasm, this is a nucleus. A protein will bind to CRIM1 that has a nuclear export signal. It will get translocated through the nuclear pore. At that point, the cargo is released, and then the whole thing cycles over again. And that leptomycin B compound that I mentioned blocks that interaction specifically. It prevents CRIM1 from binding to any nuclear export signal. What we have found is that when we take CALM AF10, shown here, and we try to make leukemias in mice, we are able to make leukemias. If we put a mutation in 2 amino acids of that nuclear export signal, you're no longer able to make leukemias. And if you take that nuclear export signal alone and you fuse it to AF10, you now get leukemias again. So this suggests that the CALM nuclear export signal is necessary and sufficient for transformation. And what it suggests is that something involving CRIM1 is important. We found that leptomycin B prevents COLM-AF10 from associating with the HOX gene locus, and also decreases HOXA gene expression in these COLM-AF10 leukemia cells. And in gray is COLM-AF10 alone, in black is COLM-AF10 with leptomycin B, and this is HOXA gene expression. So this suggests that there's something pretty specific about HOXA gene expression and the interaction between COLM and CRIM1 that potentially can be inhibited. And this takes place after about 1 hour of exposure to this drug. So it doesn't have to do with protein synthesis, doesn't have to do with messenger RNA half-life, it's a direct inhibition. So this suggests that CRIM1 plays a critical role in targeting COLM-AF10 to the HOX gene locus, and the question is how. Our first aim is to look at mechanisms of and the role for CRIM1 in regulating these HOX genes. This is something that has not been described before. CRIM1 is known to be a nuclear shuttler, basically shuttling things out of the nucleus, but a direct involvement with HOXA genes has not been reported. And because HOXA gene expression is involved in many leukemias, this may have implications beyond just this relatively rare subset. This is a sort of blow-up of that nuclear pore, and it shows some of the proteins that are involved. Up here is CRIM1 bound to a nuclear export signal containing protein. And what's important here is that there are a couple of other proteins that are parts of the nuclear pore, both of which have been identified in other translocations in leukemias. And they presumably have some role, but what role that is not clear. One of these in particular, called NUP98, is a part of the nuclear pore, but it can actually migrate off the nuclear pore and actually into the nucleus itself. And there's some suggestion that this may be involved in HOXA gene expression through mechanisms we don't understand yet. That's part of the goal of this first aim, is to look at the interactions at HOXA genes with CRIM1. But taking it back to the patient, CRIM1 inhibition is clinically translatable. I mentioned leptomycin B. It was actually used more than 20 years ago in a clinical trial because of some thought that it may be effective. But the clinical trial was halted early because of very significant toxicity. It turns out that leptomycin B blocks all nuclear export, and so proteins get trapped in the nucleus, and it really messes up cells. But if, as we postulate, there's a novel mechanism, a novel interaction of CRIM1 with HOX genes, that can be potentially inhibitable by leptomycin B or other compounds, then we could potentially have a more directed effect. And our second aim is to use CRIM1 inhibitors in vitro and in vivo mouse models. And this would provide a further rationale for clinical trials of CRIM1 inhibitor use in pediatric patients. We're collaborating with a company called Karyopharm, which was designed, Karyos comes from the word for nucleus, and this company has actually taken leptomycin B and made a bunch of modifications so that they make nuclear export inhibitors or CRIM1 inhibitors that are much, much less toxic. They've been used in clinical trials in leukemias, in many, many different solid tumors, and they have incredible potency and very minimal side effects. So we're working with this company to test these in our models, which can then potentially be translatable to patients. So I'll finish by again thanking Hyundai for the incredible support they've given to us and for their support of pediatric cancer research as a whole. Thanks.